Okay, so let's now start with the retina. Okay, so what is the retina? The retina is a thin transparent membrane that is present between the vitreous and the choroid. So it is placed between both the vitreous internally and the choroid externally, extending from the edge of the optic disc to the ora serrata, where it becomes fused with the ciliary body. So as we said in the first lecture, it is the inner nervous layer or inner neural layer of the coats of the eye, which is placed between the vitreous internally and the choroid here externally, extending from the edge of the optic disc until they reach the ora serrata, which will become fused with the ciliary body. So if we have a section here in the wall of the eye, what can we see? From inner to outer, we have this is, let's start from outer to inner. The outer layer will be the sclera, as we can see here. Then we have the vascular layer of the choroid. Then we will reach to the retina. So the retina will be formed of two main parts. The first part is called the retinal pigment epithelium externally, and internally it will be formed by the sensory retina. Here we are. So this is the retinal pigment epithelium, and here are the sensory retina. So the retinal pigment epithelium, it is just a single layer, while the sensory retina is about 9 layers. So here we can see these are the sensory retina, or the 9 layers of the sensory retina, resting on the retinal pigment epithelium, while this is the choroid, and finally we reach to the sclera. Histologically speaking, we have 10 layers of the retina. From external to internal, we have the retinal pigment epithelium. Then we have the photoreceptor layer, then the outer limiting membrane, the cell bodies of the rods and the cones, then the outer plexiform layer, inner nuclear layer, inner plexiform layer, ganglion cell layer, and nerve fiber layer, and finally the internal limiting membrane, which will separate the retina from the vitreous body. Again, these are the 10 layers of the retina, from external to internal: retinal pigment epithelium, photoreceptor layer, outer limiting membrane, outer nuclear, outer plexiform, inner plexiform, ganglion cell layer, nerve fiber layer. So by studying these layers, by knowing the characters of the cells of each layer, and by knowing the different regions of the retina, we will be able to study the anatomy of the retina. So just bear in mind that the light, when entering the eye, it is not entering from outside, but entering from inside. So it will strike the retina from the side of internal limiting membrane, and it will pass through all layers of the retina until it is perceived by the retinal pigment epithelium and the photoreceptors, where the visual cycle will occur, as we will mention in the physiology. So let's start first with the anatomy of the retinal pigment epithelium. So the retinal pigment epithelium, it is a single layer of 5 million cells, and this number is important because it is sometimes asked in the exams. It is firmly adherent to the basement, its basement membrane, which is the Bruch's layer, extending from the optic disc posteriorly to the ora serrata anteriorly, as well as the rest of the retina. The functions of retinal pigment epithelium will be studied thoroughly in the physiology, but just to have an idea about the functions are that it has excess melanin pigments, and this melanin will cause absorption of the scattering light within the eye, and it will cause the support to the photoreceptor outer segment during the visual cycle, and again causing breakdown of these photoreceptor outer segments during the visual cycle, and takes part in the recycling of vitamin A and the transport of nutrients from choriocapillaris to the photoreceptors. So here are the photoreceptors, and here are the retinal pigment epithelium. The Bruch's membrane act as its basement membrane, and here are the choriocapillaris. So The retinal pigment epithelium will cause will allow transmission of nutrients from the choriocapillaris to the photoreceptors, and also it has excess melanin content, so it will absorb the excess light and it will integrate with the photoreceptor outer segment, causing their breakdown during the visual cycle in order to maintain the vision. So it is formed by, or the structure of the retinal pigment epithelium, it is formed by a basement membrane, which is firmly adherent to the Bruch's membrane, and the pigment epithelial cells. So as we said, the pigment epithelial cell, it is a single sheet of 5 million cells of hexagonal cells. So from our previous lecture, do you remember another cell of hexagonal type that we said in the previous lecture? Yes, that's right. It is the endothelium of the cornea. So the retinal pigment epithelium is a hexagonal cells arranged in a cobblestone arrangement, as you can see here. So this is the choriocapillaris, and this is the Bruch's membrane, and here we can see the retinal pigment epithelium. So in the cross section, this is the like a flat section. But if you have a cross section to see the cells of retinal pigment epithelium in more details, you will find that they are tall and slender in the center, just beneath the macula and the fovea. However, if you are going more periphery, they are becoming more cuboidal. This is very important because this will, the being tall and slender will enable the central retinal pigment epithelium to be closely in contact with the light that hitting these central cells and central cones, as we can see later. So let's have a closer look on the retinal pigment epithelial cells. At the base of the retinal pigment epithelium, you will find that there are prominent basal enfoldings like this. This is just the basal enfoldings of the plasma membrane. You will be able to find many mitochondria like this. But the pigments or the melanin pigments is not present at the base of the cells, and this is also asked in the exam. So the pigment, as you can see here, it's present near the apex, not near the base. Then we have the body, and the body will contain a large nucleus, can contain some organelles like Golgi apparatus, as you can see here, like endoplasmic reticulum, as you can see here. There may be some lipofuscin granules, and these granules will be responsible for the yellowish color of the fovea, and the melanin will start to appear at the body. Then we have the apex, and the apex will show like finger-like projections or villi. These villi are interdigitating with a disc-like structure called the photoreceptor outer segment. So this is the photoreceptor outer segment, and this is the villi of the retinal pigment epithelium. So as you can see that they are both integrated together, and this means that they are both functioning together for the purpose of visual cycle. The apical cytoplasm will contain more melanin, as you can see here, and sometimes or almost always they will present digested outer segment photoreceptors. So in order for visual cycle to occur, this outer segment's photoreceptor will be digested within the retinal pigment epithelium. That's why they are both integrated together like this. And once the outer segment photoreceptor is ingested within the retinal pigment epithelium, it is called a phagosome. Phagosome. The lateral walls of the retinal pigment epithelium are joined together by specialized cell junction formed of zonular adherents and zonular occludence. So this is again, like we said in the cornea, these are cell junction or specialized cell junction that will cause a restriction of movement of large size molecules between the cells, and this is in the retina. It is present between the lateral wall of the retinal pigment epithelium and not the apex. So this also has been asked in the MCQ before. So remember that the terminal bars or the zonular adherents and zonular occludents are present between the lateral walls of the retinal pigment epithelium, not the microvilli, or not these microvilli. And this is in turn form what's called the outer layer of the blood-retinal barrier. So as we said that there is a blood-aqueous barrier in the previous lecture, and we will be discussing it in more details in the physiology. We'll just know the anatomical part here, which is the of the blood-retinal barrier, that it is a barrier present in the retina preventing large molecules from entering the retina, such as microorganisms or antibiotics or plasma proteins, so as this will impair the transparency of the retina. So this blood-retinal barrier is formed by outer part and inner part. The outer part of the blood-retinal barrier is the tight junction or the zonular occludence between the lateral walls of the retinal pigment epithelium, and the inner blood-retinal barrier will be formed by the tight junction between the non-fenestrated endothelial cells of the retinal capillaries. So these together will form what's called the blood-retinal barrier. So again, here is the tight junction between the lateral walls of the RPE and the tight junction between the endothelial cells of the retinal endothelium. Both of them will act as a barrier to prevent movement of unwanted molecules to enter the retina. Again, these are the retinal pigment epithelial cells, and this is the tight junction between the lateral walls of the cells, and this is the tight junction between the retinal endothelial cells. Both of them will form what's called the blood-retinal barrier. Okay, so let's now move to the anatomy of the neurosensory retina, or the inner 9 layers of the retina. And this can be classified into some, we will say first the retinal cells that will form the retina. Then we will arrange these cells into layers, so we will know what are the 9 layers of the neurosensory retina, and and every layer, what are the cells that will consist each layer of the retina. Then we will discuss the retinal regions, some important retinal regions with specific anatomical details. So the first cells in the retina are the rods and the cones, and this collectively are called the photoreceptors. The number here again is important because we have 120 million rods and only 6.5 million cones. So as you can see, the rods are more predominant and more numerous than the cones. These are the rods and these are the cones. So if we have a section here in the retina, you can see that. Just hold on for a second, please. Okay, sorry for interruption. So we have here are the retinal pigment epithelium, and here are the rods and cones. So The rods here are interdigitating with the microvilli of the outer of the retinal pigment epithelium as well as the cones. But as you can see, that there are from the first instance, you can see that there are huge difference in the shape between both rods and the cones. So is this the real fact? The answer is yes, because the rods are more cylindrical in shape and more tall than the cones, while the cones are shorter and more conical. So, what are other differences? Let's see. Let's first divide them into outer segment and inner segment. The outer segment, which is the part of the photoreceptors that is interdigitating with the retinal pigment epithelium. So, this part is formed of discs, and these discs are what carry the visual pigment, which is responsible for the visual impulse. So these discs are present in the outer segment of both the rods and the cones, but with a great difference that in these discs are separated from the plasma membrane in case of the rods, while the discs are continuous with the plasma membranes in case of the cones. So what is the other difference is it is that the outer segment or the discs of the visual of the rods contain the rhodopsin, which is responsible for night vision. So that's why the rods are responsible for night vision, while the cones are carry the color photopigments. While the cones are carrying the color photopigments, which is responsible for the day vision and the color vision. After the photoreceptor outer segment, then there will be a filament connecting the outer segment to the inner segment, which is called the cilium. So the cilium will connect the outer segment containing of the discs and the inner segments. So the cilium will connecting the outer segments containing the discs and the inner segment, which has the mitochondria and the nuclei and other organelles. And if we have a cut section here within the cilium, it will be present like 9 pairs of microtubules, as you can see here. So this is just a cut section of the cilium at this area. There's just 9 pairs of microtubules, and its main function is to support the growth and of the discs from the mitochondria and from the nucleus. So it is just a connecting stock between the outer segment and the inner segment. Then we have the inner segment, and the inner segment will be consisting of the outer portion and the inner portion. The outer portion of it is like here. Sometimes it can be called refractile portion or ellipsoid portion. And the inner portion is called the non-refractile or the myeloid portion. The outer portion, it is mainly its main function of the outer portion of the inner segment is in energy production. That's how can be maintained by the numerous mitochondria present at the outer portion of the inner segment. While the inner portion, it is like here. The inner portion containing visual pigments, ribosomes, Golgi apparatus, vesicles, and vacuoles. So these are usually, or the main function of the inner portion is synthesis. So they are responsible for synthesis of the visual pigments. So again, the inner segment is consisting from an outer part and the inner part, like here. The outer part containing mitochondria, which is responsible for energy production, and this energy is needed by the inner segment or the inner portion for synthesis of the visual pigments, and also it has different cellular organelles such as Golgi apparatus and ribosomes. Specifically, another part of the outer fiber and the inner fiber, or the outer portion, inner portion, and there will be what's called outer fiber. And the outer fiber, it is a port into the inner segment of the photoreceptors, which connect the outer and the inner portion to the nucleus. So this part is longer in the rods because the rods are longer, while it is shorter in the cones because the cones are shorter. And the very important part about it is that it is joined horizontally by by what's called the terminal bars of zonal adherence of molar cells, which form external limiting membrane. Meaning that you can see this is the section of the retina. So these are the choriocapillaris, these are the retinal pigment epithelium, and these are the photoreceptors. So here are the nucleus, and here are the outer portion. Or the outer fiber. So the outer fiber here, as you can see that the outer fibers of both rods and the cones are joined by a horizontal structure, this dark line. And this dark line is a part of a vertically oriented, very tall cell called the molar cell. So this horizontal line, and it's called the zonal adherence or the cell junction by the molar cell. It's called zonal adherence, and it is joined to the outer fiber together of so both of them together are called the external limiting membrane, which forms one of the layers of the retina. So again, outer fibers are together with the zonal adherence of the molar cells form what's called external limiting membrane. Here is the molar cells, and here is the external limiting membrane formed by the zonal adherence of molar cells and the outer fibers. So the outer fibers of the photoreceptors. Then we will come to the cell body. So the cell body of the photoreceptors are forming a layer of the retina. It's called the photoreceptor layers. So this layer are located into the outer nuclear layer. So this layer or it's formed by the nuclei of the photoreceptors, either the rods or cones. Then finally the inner fiber. So this is the outer fiber and this is the inner fiber. The inner fiber connecting the nucleus to the terminal expansion of the photoreceptors. And this terminal expansion is different according to the type of photoreceptors. So we have two types. We have what's called the rod spherule, which is the terminal expansion of the rods, and cone pedicle, which is the terminal expansion of the cones. Both of them will synapse in the outer plexiform layer with those cells whose nuclei are present in the inner nuclear layer, which are the bipolar cells and horizontal cells and the bipolar and horizontal cells, and also the molar cells. So here we can see that the anatomy of the rods and the cones. The rods is formed by the outer segment, which is formed by photoreceptor outer segment interdigitating with the with the villi of the retinal pigment epithelium containing discs. Then we will have a cilium that will connect the outer segment to the inner segment. Then we will have the inner segment, which will consist of outer portion and the inner portion, and then outer fiber, and the outer fiber will be forming the external limiting membrane with the zonal adherence of the molar cells. Then we will have this is the outer fiber here, and then we will have the cell nuclei forming the outer nuclear layer of the retina. Then we will have the inner fiber connecting the nucleus to the terminal expansion, which can be either rod spherule or cone pedicle. And this cone pedicle and rod spherule are forming sharing in the formation of a layer called outer plexiform layer. So, what are other cells than the retina, other than the retinal pigment epithelium? You only need to know the retinal pigment epithelium in such details. While other cells, it is just enough to know where are the cells located and where are their dendrites or cell process are located. So, here we see that these are the photoreceptor cells. So the nuclei will form the outer nuclear layer, and their rhodospherule and comb pedicle will synapse with other cells. These cells, their nuclei are present into the inner nuclear layer, which are the bipolar and horizontal cells and the molar cells. So the bipolar, meaning by saying the bipolar, so it has two poles. These cells have their cell body present into the inner nuclear layer, while their dendrites are present into the outer plexiform layer. So again, the nuclei are or the dendrites are present into the outer plexiform layer, synapsing with the photoreceptor cells. Their nuclei are present into the inner nuclear layer, as you can see here, and their synapse also with at the inner plexiform layer with the retinal ganglion cells and the amacrine cells. For the horizontal cells, they are located horizontally. So their axons and dendrites, both of them are present into the outer plexiform layer, while the cell body it is present into the inner nuclear layer. So these are horizontal cells, as its name implies. The mirror image of the amacrine cells is or of the horizontal cells are the amacrine cells. So the amacrine cells, its nuclei present into the inner nuclear layer, while the process are present into the inner plexiform layer. So are there only these are only the only cells present into the inner nuclear layer? It is only the bipolar and horizontal and the amacrine cells? No, the answer is no, because we have another cell which is a very large cell, very tall cell, taking forming the structural framework of the retina as it extends along the whole depth of the retina. It's called the molar cells. Molar cells nuclei are present also in the inner nuclear layer, while they have outer process and the inner process. The outer process we have just mentioned that it forms the external limiting membrane with the outer fibers of the photoreceptors. You remember we said that the terminal zonal adherence of the molar cells with together with the outer fibers of the photoreceptors will form the external limiting membrane, while its inner process will form the most inner layer of the retina, which is the internal limiting membrane. So again, the molar cell have their nuclei present into the inner nuclear layer, while the outer process will form the external limiting membrane, and the inner process will form the internal limiting membrane. The final cells present in the retina are the ganglion cell layer, and since the ganglion cell layer are of extreme importance, they form a layer by its own. It's called the ganglion cell layer. So the ganglion cell layer contains the ganglion cell nuclei, while their dendrites will synapse at the inner plexiform layer with the process of horizontal cells, amacrine cells, and horizontal and bipolar cells. So these are synapses forming the inner plexiform layer, and the cell body will present at a specific cell type, a specific layer called the ganglion cell layer, and the axons of the retinal ganglion cells are the ones which will form the nerve fiber layer. So the nerve fiber layer will be the most the layer or the eighth layer of the neurosensor retina before these optic nerve fiber will collectively form the optic nerve fibers. So again, here are the ganglion cell, their nuclei present in the ganglion cell layer. Their dendrites will synapse with the amacrine cells and the bipolar cells, and their fibers will extend to form the nerve fiber layer of the optic nerve, which will emerge from the whole retina to arise from the optic disc and to leave the eye through the optic nerve. So where then it shall goes? After the fibers from the optic nerve, after the nerve fiber layer from all ganglion cells arise from the retina, they will go to the optic nerve to leave the eye. So it will leave the eye to reach either one of the two destinations, either lateral geniculate body here or the midbrain into the pretectal nucleus. So what will govern the exit? So which type of fibers will leave the eye toward the lateral geniculate body, and which type of fibers which will leave the eye toward the pretectal nucleus? This will depend whether these fibers are carrying visual impulses or pupillary response. So if the fibers are for vision, then they will be interpreted by the lateral geniculate body. However, if the fibers are for pupillary response, so they will just control the size of the pupil, they are just a sensation of the amount of light entering the eye with no visual significance, then they will not enter the lateral geniculate body, but they will leave the optic tract to synapse into the pretectal nuclei of the midbrain. And we have mentioned this in the last lecture in the pupillary light reflex pathway. So just to complete the picture today, these fibers are just the continuation of the nerve fiber layer, which are the fibers by the ganglion cell layer. So these are the cells. Is everything clear up till now? Okay. So to summarize, we have now the 9 layers of the neurosensory retina. If we add them to the layer of retinal pigment epithelium, then they will be total of 10 layers. So let's just see. These are the 10 layers retinal pigment epithelium. Then we have the photoreceptor layer. Then we have the external limiting membrane, as we mentioned before. Then the outer nuclear layer formed by the nuclei of the rods and the cones. Then the outer plexiform layer. Then the inner nuclear layer. Then the inner plexiform layer. Then ganglion cell layer. Then nerve fiber layer, and finally the internal limiting membrane. So to know, just to summarize the content of each layer, so the retinal pigment epithelium, photoreceptor, external limiting membrane doesn't have any other contents. Outer nuclear layer contain the nuclei of the photoreceptors. So just I will try to stress on this because this is very important and asked frequently in the exam to tell you what are the layers and what are its contents. So the outer nuclear layer containing the nuclei of the rods and cones. The outer plexiform layer contains synapses. Here we can see the outer plexiform layer containing synapses between the photoreceptors, which are the rod spherule and the cone pedicles, the dendrites of the bipolar cells, and the process of horizontal cells. So you see the bluish one, which are the photoreceptors, the reddish one, which is the bipolar, the brownish one, which is the horizontal cells. The synapses of these cells together with the molar cells, you can see that the molar cells are very large cell in the background, extending from along the whole depth of the retina, forming the structural framework of the retina. It's present in all layers. So don't ever forget the molar cells are present in all layers. So these are the content of the outer plexiform layer. Then we have the inner nuclear layer containing the nuclei of the bipolar cells and molar cells, the horizontal cells, and the amacrine cells. Just bear in mind that the horizontal cells are present at outer portion, while the amacrine cells are present into the inner portion. So all nuclei of the retina are present into the inner nuclear layer except what? Except the nuclei of the photoreceptors and except the nuclei of the ganglion cell layer. Then we have the inner plexiform layer, which contains synapses between the bipolar cells, the red one, the amacrine cell, the green one, and the ganglion cell dendrites, the yellowish one. And also there are the molar cell fibers are present into the inner plexiform layer. So again, it contain it join together the bipolar, the ganglion cell, and the amacrine cells, and the presence also of molar fibers. Then we have the ganglion cell layer. The ganglion cell layer, it is a single layer thick. However, it is in the parafoveal region, it is 5 to 10 layer thick. And you may wonder why. This is because it is absent in the fovea. So is this absence due to actual reduction of the ganglion cell layer? That's what we will see later on. Then the final layer, or the pre-final layer, it is the nerve fiber layer, which is formed by the unmyelinated axons of the ganglion cells arranged into bundles that are parallel to the retina, most thick around the margins of the optic disc, and their thickness is different according to the quadrant, as we said in the previous lecture. So it is the thickest into the inferior quadrant, then the superior quadrant, then the nasal quadrant, and finally the most thin will be the temporal quadrant. The arrangement of the axons of the nerve fiber layer take a specific configuration. So if this is called the optic disc, and this is the horizontal line, and here are the fovea and the macula. So the nerve fiber layer arising from the ganglion cell present into of the macula will emerge directly from the macula toward the nasal edge of the optic disc, and this will form what's called the papillomacular bundle. So again, the nerve fiber layer of the ganglion cells will emerge directly to the optic disc, forming what's called the papillomacular bundle. While the nerve fiber layer, or the nerve fibers originating from the ganglion cells temporal to the disc, but not from the macula, will just arise above and below the central fiber, or arise above and below the macula without crossing the horizontal raphe, or without crossing the horizontal line. So the temporal fibers again will arising from the ganglion cells, the temporal to the disc, but not from the macula, will arch above the macula above, and arch below the macula below, but not crossing the horizontal line, and to finally end into the optic disc. While the nasal fibers originating from the ganglion cells and nasal to the optic disc are extending directly toward the optic disc. So they don't arch, they just extend directly to the optic disc. So these are arrangement of the axons in the nerve fiber layer of the retina. I hope it's clear, and sometimes it is asked in the exam. The final layer, or the most inner layer, it is called the internal limiting membrane, which is formed by the outer layer and inner layer. The outer layer is formed by the foot plates of inner process of the molar cells. So here is the internal limiting membrane, which is formed, as you can see, by the molar cells. So the molar cells have a foot plate. This foot plate is forming the most inner layer of the retina, while the inner layer, it is just a basement membrane. Finally, we come to the retinal regions. There are some certain regions in the retina with specific anatomical description and anatomical details. So let's see them. We have the first, the ora serrata, and this is the peripheral retina. And here is the central retina, or what's called the macula lutea. So what is the ora serrata? We have been using this term since the last lecture, since the previous lecture, and now we come to know what is the ora serrata and to know what is the anatomical description of it. So basically, the ora serrata, it is just the termination of the retina. So the termination of the retina, it is not in like a straight line, but it is a scalloped line. So it is a scalloped anterior termination of the sensor retina just adjacent to the pars plana. So you can see here is the sensor retina, and you can see that there is a dentate-like line, or the line which has a corrugations. These corrugations are called the ora serrata. So where is the ora serrata is located? It is located about 8.5 millimeter from the limbus, as you can see here, while it is located about 6 millimeter anterior to the equator of the eye, and it's also located about 25 millimeter from the edge of the optic disc. So these numbers are important. It is present 8.5 from the limbus, 6 millimeter from the equator, and 25 millimeter from the edge of the optic disc. The shape is like dentate process as we see here, and these dentate processes are best prominent or best marked and more prominent at the superior nasal quadrant. Remember the superior nasal quadrant. So we said in the last lecture the superior nasal quadrant has a specific importance also when we mentioned about the most area liable to rupture in the sclera, which will be the superior nasal quadrant. So remember again that the dentate process is more marked at the superior nasal quadrant. So it is attached at the ora serrata. The retina is firmly adherent to the choroid under it and the vitreous in front of it. And the sensor retina is firmly adherent to the retinal pigment epithelium. This is again of anatomical importance. So in the ora serrata, all structures are adherent to each other. The retina is adherent to the neurosensor retina is adherent to the retinal pigment epithelium, and the ora serrata, the retina is adherent to the structure underlying it, which is the choroid, and the structure overlying it, which is the vitreous. So here we come to another transition slide, and as we said in the last lecture, the transition slides are important and they like to ask about it in the exam. So what are the changes that occur at the ora serrata? So the retinal pigment, all changes you will notice are due to loss of function, meaning that we don't need the peripheral retina are not a visually significant area, meaning that we don't use the peripheral retina for vision or for visualizing details in our day-to-day life. So all structures in the retina of the neurosensor retina, which are important for maintaining a sharp vision, will start to decline. For example, the retinal pigment epithelium, we don't need it anymore, so it become more large and more irregular with variable amount of melanin. The photoreceptors, the rods are they start to disappear first, but the rods are more evident in the peripheral retina than the cones, so they will become, although they are numerous, but they start to decrease in number and disappear first, while the cones are no longer present. So the cones don't reach the peripheral retina because they are responsible for central vision, daylight vision, and the color vision. So they are present also in the macula. So you may just find some malformed cones, and they disappear rapidly. The nuclear layer, outer nuclear and inner nuclear layer, fuse together and become more thin in order to prepare to form the pars plana. Ganglion cell layer becomes scattered and arranged in only single layer. The external limiting membrane will continue between as a basement membrane of the ciliary epithelium, as we said in the previous lecture, and the internal limiting membrane will continue as the basement membrane of the non-pigmented ciliary epithelium, as we again said in the previous lecture. Then finally, we have the central retina, or what's called the macula lutea. So lutea meaning yellowish. So the macula has a yellowish hue color, and this is just an oval area within the central retina. So the central part of the retina, it is oval in shape. It is called the macula lutea. This macula is occupied by the central cones, and that's why they are responsible for day vision and the color vision. So the thickness of the macula altogether it's about 350 micron, while the thickness of the macula at the fovea it is only 90 microns. And you will see now why is this reduction of thickness. Let's now just talk about the extension of the macula. So the macula is occupying the central part of the posterior part of the retina, which is about 5 millimeter diameter. It's a central round or oval area of about 5 millimeter in diameter in its vertical diameter and its horizontal diameter that lies within the temporal arcades. This is called the superior temporal arcade, and this is called the inferior temporal arcade. The area between both of them, it is called the macula lutea. We said in the previous lecture that there is a relation between the fovea or the center of the fovea to the center of the optic disc. So the center of the fovea, it lies about 3 millimeter temporal to the optic disc edge, while it lies more inferior to the optic disc. So it is only 1 millimeter below the optic disc center. Again, the fovea is 3 millimeter temporal to the optic disc edge, while it is 1 millimeter below the optic disc center. So this is the macula, and the macula for itself this is the fovea. Number one, it's the fovea, and the areas of surrounding or the fovea itself has different anatomical subdivisions. So this is a flat section and this is a cross section. So you can see the center of the fovea is called the fovula, which is the area of sharpest vision that we can perceive. It's about 0.35 millimeter in diameter, and it has a central concave indentation within the fovea. As you can see here, this is what's called foveal contour. The foveal contour is concave, and this is normal. And when you are examining it, if it is healthy, it will sometimes show a central light reflex. It's called the ambu. So the ambu is just a central light dot present at the fovula. At a diameter of about 1.5 millimeter, this is called the fovea. So the fovea here is all called the fovea. The fovea is about 1.5 millimeter in diameter, and it is a central depression within the macula. So if we said that the macula is 5 millimeter in the vertical diameter and 5 millimeter in the horizontal diameter, the central 1.5 millimeter, it is called the fovea. Around the fovea in zone 3, it is called the parafovea. So the parafovea, it is an 0.5 millimeter area wide that encircles the fovea, while the perifovea, it is about 1.5 millimeter wide area that encircles the parafovea. So why do we have these subdivisions? Because this will have different anatomical and histological configuration. Again, a slide of transition. So what are the changes that occur at the macula? That's why we are studying these specific regions, either the ora serrata or the macula. These are of anatomical importance because there are certain anatomical changes that occur at these layers. So for the macula, all changes will be the reverse from the ora serrata. So all the changes should serve the higher visual function of the macula. So they should receive better nutrition. They should have more density of the photoreceptors, and they will show they have to show better retinal transparency, as we will see now in the arrangement of layers. So Choriocapillaries will do their job by providing more dense capillaries, more wide and more large fenestration to support the nutrition of the photoreceptors and the retinal pigment epithelium. So the retinal pigment epithelium and the photoreceptors are supplied by the ciliary circulation coming from the choriocapillaries. The retinal pigment epithelium, there will be only cones present in the fovea, and this will serve to be the highest density in the retina. So the highest density of the cones in the retina will present in the fovea to support the central sharp vision. So this is the area which is responsible for target fixation. So if you are fixing on a target, the image of this target will fall onto the fovea and will fall on the central cones. So this central cones should have specific characters. These are to be densely packed. They will be tightly packed together. There will be more tall, more thin, so that we will be able to provide higher density of the central cones. The rods are not present in the fovea, but they are just present in the macular periphery. The external limiting membrane has holes, as these holes will act as a passage for the inner segment of cones. And since the cones present in the macula or in the fovea are thin, so these holes will be thin as well. Sometimes, due to greater density of the cones, this will lead to pushing the external limiting membrane forward, and we will see this later on. And sometimes this can be referred as the fovea externa. So the fovea externa, meaning that there is a tightly packed cones into the central retina or the central of the macula. So these tightly packed central cones will cause pushing of the external limiting membrane forward. So it acts like as if it is another fovea with its concavity. It's called the external fovea or fovea externa. Outer nuclear layer, it is formed by the nuclei of rods and the cones as we know, and sometimes you may be able to find some extruded nuclei which are aberrant nuclei, so they are not present into the outer nuclear layer, but they are just present at the outer part of the external limiting membrane. So I will just, okay. The outer plexiform layer, it's formed by synaptic junction between the rods and cones bipolar and horizontal cells, while the inner nuclear layer, it is containing the nuclei of bipolar, horizontal, amacrine, and molar cells, as we said before. So this is there are nothing new here in the outer plexiform layer and the inner nuclear layer. The inner plexiform layer containing the synapses between the bipolar and bipolar axons, ganglion dendrites, and you remember that solitia, meaning that it is yellowish, and yellowish due to the presence of xanthophyll pigments, which is mainly present into the ganglion cells. And here we come to the striking fact. So sometimes they ask you the exam that in the fovea, the ganglion cell will show increased number in layers. And you may think about, you may think of it by logic that this is right, because the ganglion cell layer is important for vision, so they should be present at large volume in the fovea. And the answer is wrong, because they are absent. The ganglion cell layer is absent in the fovea, and this has an important anatomical and optical value. This is because we need the fovea to be as thin as possible, and that's how the light will be striking the photoreceptor directly. So other layers than the photoreceptors and the external and the internal limiting membrane will be pushed to a side. So the ganglion cell layer, as you can see here, are pushed to a side. The bipolar cells are pushed to a side. So this will lead to that the light will enter the fovea, will be striking the central cones directly. So it is arranged into two or more layers Usually, the ganglion cells in the macula are arranged into 2 or 3 layers, as you can see here. But in case of the fovea or the central fovea, it is absent. The nerve fiber layer is for at the area of the macula is forming the papillomacular bundle, as we said earlier. It is and it will pass directly toward the nasal edge, or sorry, the temporal edge of the papillomacular of the optic disc. And the temporal fibers do not pass through the macula. As we said, the temporal fibers arch above the papillomacular bundle and below the papillomacular bundle, and they don't cross the horizontal midline. The internal limiting membrane, it is the thickest part of the thickest area of the internal limiting membrane will be present at the macula. So these are the changes. Just a specific or more hints about the fovea. So the fovea here, as you can see here, this is the foveal depression or the central foveal depression, which is a concave-like depression. It is the fovea is formed of only 3 layers. These 3 layers are the photoreceptor layer and the internal and the external limiting membrane. So this thinning, it is making the fovea will be about 90 micron. On the other hand, the macula is about 350 microns, and this decrease in the thinning, it is not due to actual reduction. This is just due to displacement of the cells of the bipolar and the ganglion cell layer and all other layers to a side. So this will make that this thinning is not due to actual reduction, but due to displacement peripheral, as you can see here. And this displacement will lead to formation of what's called the Henle layer. So what is the Henle layer? Henle layer, so you can see here is the rods and cones, and here is the outer nuclear layer, and you can see that there is an increase in the density of the central cones, making the outer nuclear layer so that they will be directly exposed to the light. Why there was a layer beyond the outer nuclear layer, the next layer beyond the outer nuclear layer, it's called the external outer plexiform layer. The outer plexiform layer, due to the increased density of the central cone, will be pushed to a side, as you can see here. So this pushing of the external limiting of the outer plexiform layer will lead to radially arrangement of the fibers of the outer plexiform layer. So they are becoming more oblique and more horizontal than more horizontal and more oblique inner fibers. So we know that it's formed by the inner fibers of the photoreceptors. So, but it is the only thing that it become more oblique and more horizontal, not vertically oriented, but become more oblique. So they appear as if they are radiating from the fovea. So it is very common question in the exam. What is the Henle layer? So the Henle layer, it is just the outer plexiform layer that is formed from the inner fibers of the photoreceptor, that they just come to become horizontal and oblique due to pushing of the central cones or the increasing density of the central cones. And then the clinical correlation of the Henle layer is sometimes due to exudation. For example, there are some diseases that will cause exudation. If these exudation are present into the Henle layer, they will take the configuration of the Henle layer, forming what's called macular star. So macular star is basically a macular exudation in which the exudates or the hard exudates are following the arrangement of the Henle layer. This can occur at some diseases like neuroretinitis or malignant hypertension. Since the cells of the bipolar and the ganglion cells layer are pushed from a side in the fovea, they will be accumulating into the perifoveal region. So the parafoveal region. So the parafoveal region will show the highest density of the bipolar cells and the ganglion cells. So bipolar and the ganglion cells will show become more increased at the parafoveal region. However, when we changes to the perifoveal region, so in the perifoveal region, we are now away from the central fovea. So the density of the cones decrease markedly. So you can see here at the fovea, at the area of the fovea and the parafovea, the cones are densely packed. However, when we return to the perifoveal region, the fovea or the perifovea will show decreasing the density of the cones. Also, the outer plexiform layer change from the Henle layer configuration. As you can see here, the Henle layer, the external limiting membrane of the forming the Henle layer was obliquely arranged. However, when we reach the perifoveal region, the external limiting membrane become regular arrangement, not oblique arrangement of the Henle layer. So here is what I just mentioned about the fovea externa. So in the fovea externa, meaning that there is sometimes a bulge of the photoreceptors. So bulge of this layer or bulge of the external limiting membrane here, as you can see here, this sometimes can be referred to a fovea externa, and this is caused by the external limiting membrane, which is pushed against high density of the central cone. And sometimes you may be able to find some nuclei outside the external limiting membrane, here or here or here or here, some nuclei of the photoreceptors, and these are some extruded nuclei or aberrant nuclei. Finally, the blood supply of the retina will be discussed in details in the vascular blood supply of the eye. But just to summarize, the retina receives its blood supply from the central retinal artery, which share in the blood supply of the outer two-thirds of the retina, while the inner one-third of the retina, which is supplied by branches from the ciliary circulation or from the choriocapillaris, as we said earlier. Sometimes the ciliary circulation will provide an artery called the cilioretinal artery. So the cilioretinal artery, it is present in normally in 20% of human population, and this artery will share into supply of the macula. So the macula should be supplied by the or the photoreceptors of the macula and the retinal pigment epithelium, or the layers of the macula should be supplied, the outer two-thirds should be supplied by the branches of the central retinal artery, but sometimes in 20% of people, the ciliary circulation will share information or share in supply of the central retina. So here is the picture of the healthy retina. So this healthy retina show these are the branches of the central retinal artery. These are the upper temporal, and this is the upper nasal, and this is the lower temporal branch retinal artery, and this is the lower nasal branch retinal artery. These are arteries arising from the temporal or the final divisions of the central retinal artery, which are responsible for a supplying of the outer two-thirds of the retina, while the inner one-third is supplied by the branches from the ciliary arteries. If there is a central retinal artery occlusion, this will lead to complete pallor of the retina, as you can see here. So the retina lost its blood supply. However, the lucky 20% of people which have cilioretinal artery, if these people got central retinal artery occlusion and they have patent cilioretinal artery, they will still have retinal pallor and retinal edema due to central retinal artery occlusion. However, they will have preserved central vision due to patent cilioretinal artery, as you can see here. So the papillomacular bundle will be supplied or preserved, and these people or these patients will be still have been able to maintain the central vision caused by the cilioretinal artery. So this is the end of our lecture today.